All right, in this video we will talk about the basics of the chi-square analysis and then how to conduct that in RStudio. Again, just base RStudio for the purposes of our course. Um, so again, most of you are probably not excited about working with statistics, but again, they play a critical role in the scientific process, right? We use them to summarize scientific information. Those are our descriptive statistics. We use them to make estimations or inferences about populations, and then we use them to test specific hypotheses, also known as inferential statistics, which are sort of the focal of our course, right? And so in this case, we're focusing on the chi-square analysis, which is just one of many types of inferential statistics, right? Again, keep in mind that in essentially any data set, that's a sample of a larger population, right? So here in this figure, we've got the blue shape that represents the entire population, right? Whether this is the entire distribution of potential values or this is a whole population of a certain organism, right? Generally, it's impossible to take a census of the entire population, so we're pulling samples out of that population, which is a subset, and trying to make inferences about the larger population given the subset that we've looked at. So there's always a chance that our inferences or our estimates are off, right? And so we need to keep type 1 and type 2 error in mind. So type 1 error is called the error of the first kind, or alpha error, also called a false positive, right? So false positive, it means that we're saying there is a difference when there isn't one. So it's the error of rejecting the null hypothesis when it's actually true. So basically we're saying that there's a difference in treatment, or that these two things are not equal when actually they are equal, right? Or this value is different than 0 when it actually is equal to 0, right? We set our type 1 error rate by giving you an alpha cutoff or a p-value cutoff. So when we say that we use an alpha of 0.05, that means that we're saying we're okay having a 5% chance that we've made a type 1 error. Okay. I mean, essentially the other way to view that is there's a 5% chance we get a larger difference by chance than what we have seen, right? So our alpha is what we're setting at our type 1 error rate, right? Type 2 error is a beta error, or error of the second kind, known as false negatives, right? The error of failing to reject the null hypothesis when the alternative hypothesis is the true state, right? So we're saying that they're the same when actually they're different, right? And you can think about this of any, hospital test or whatever, right? So we've survived the pandemic. COVID tests have a type 1 and a type 2 error rate, right? We have a chance of giving a false positive and a chance of giving a false negative. Those things we keep in mind as we design our experiments. When we talk about statistical analysis, you'll notice that we'll talk about the null and alternative hypotheses. Those are different from when we talk about experimental or scientific hypotheses when designing or coming up with our experiment, right? So we spend a good chunk of class talking about experimental or scientific hypotheses, right? They address a biological relationship between two or more variables. They're testable, right, or falsifiable. It's a proposed explanation for an observed pattern or trend, right? That's an experimental hypothesis. When we conduct a statistical analysis, we have statistical hypotheses. These are statements about whether or not a pattern or trend or difference is present in your data. Okay. These are just for the statistical analyses. They don't talk about the relationship and they don't talk about the variables per se, right? These are different than experimental hypotheses. Each statistical analysis has a particularly worded null hypothesis and alternative hypothesis. And that's what we're testing for when we talk about the rejecting the null or not rejecting the null, right? Those are the statistical null hypotheses versus the alternative hypothesis. Okay. So you have to remember there's a big difference between experimental and statistical hypotheses and when each one is appropriate to use. Also, the wording for statistical hypotheses is specific depending on what statistical analysis you're doing. The null hypothesis for a chi-square is not the same as it is for a t-test. That's not the same as it is for a regression or an ANCOVA, right? They have very specific null and alternative hypotheses for the analysis that you're doing, okay? And the wording needs to be appropriate given the analysis that you're doing. Okay? So again, the null hypothesis, often denoted by H naught here, or H sub 0, states that there is no pattern or trend in the data, there's no difference, there's no relationship, the value doesn't differ from 0, right? It depends on the analysis that we're doing, but basically is that there's nothing there, right? The alternative is that there is a difference, right? The value does differ from 0. The two groups are different, right? The two groups are not the same, right? So it depends on the analysis that we're doing, but the null and alternative hypothesis are what we use in the statistical analysis, and they differ from the experimental hypothesis that you came up with before we even collected the data. This is my statistics roadmap. So this helps me know what statistical analysis I need to do or I should do given the types of variables that I have, right? So here's my X, my independent here, my Y, my dependent there, and the types, categorical versus continuous. We're covering chi-square analysis. That's when we have a categorical X and a categorical Y. So I fall into this cell right there. Again, I like this roadmap. I use it quite frequently. Get in the habit of using this to tell you what analysis is most appropriate given your variables. Okay? So here I'm going to have one categorical dependent and one categorical independent, and that tells me that I need to do a chi-square. Okay? Excuse me. So again, chi-square analysis is a statistical test used to determine if observed values significantly differ from expected values, right? We use this with categorical variables, proportional data, or frequency data. Okay? Again, one X, one Y, or one dependent and one independent. The chi-square test can actually be used to test a variety of things or look at a variety of things. We can use it to see if there's a goodness of fit, so to look at distributions, right? Test of independence, test of homogeneity, right? But what we normally use it for is to compare the independence of two characteristics. Is one characteristic dependent on the other? Okay? So again, chi-square can be used to ask a variety of questions, but the most common that you'll see in this course is using it to compare or to see if two variables are independent of each other. Okay. Again, the chi-square is denoted here, and it's the summation of our observed minus our expected squared divided by our expected. So we sum up on our observed values minus our expected values and square that, divided by our expected, and we sum that across all of our cells, and that gives us our chi-square value. Again, the null hypothesis is that the observed values are not different from the expected, right? So observed equals expected, or in the case that the question we're actually asking, variable 1 is independent of variable 2. Okay? Because we're testing for independence. The alternative hypothesis is that the observed does not equal the expected, or that variable 1 is dependent or influenced by variable 2. So if we look at an example here, so here's, if we look at whether you identify as a Democrat or Republican and your genetic sex, X, Y, or XX, right? And we just count, so let's say we go survey 100 people, 50 of them are XY, 50 of them are XX, and we say, do you consider yourself a Democrat or Republican? And these are the results. 20 XY said Democrat, 30 said Republican, 30 XX said Democrat, 20 said Republican. Okay? So these are our observed values, right? What's the null and alternative hypothesis for this data set in particular, right? The null hypothesis is that political affiliation is independent of genetic sex. Excuse me. And the alternative is that political affiliation is dependent or influenced by genetic sex, right? Next, we need to calculate the expected values for each cell. This was pretty easy. We got two choices, Democrat or Republican, two genetic sexes, 100 total, right? So we would expect, if there was no relationship, that 25, that would be 25 and 25, 25 and 25, right? Now we go through and calculate our chi-square, observed minus expected squared minus, or divided by expected, and we sum that for our 4 cells, right? So 20 minus 25 squared divided by 25, 30 minus 25 squared divided by 25, okay? It actually gets me a value of 4, right? So the chi-square value is 4. Now I need to know the degrees of freedom. And we can, it's equal to k minus 1, whereas the k is the number of groups or cells within a category. N minus 1, where n is the sample size. Or rows minus 1 times columns minus 1. They should generally get you to the same place. Okay. And then this is what it would look like in RStudio, where they would give you the chi-square value, the degrees of freedom, and our associated p-value. Okay. This is for a different data set. This would be for the data set here with the genetic sex and political affiliation. Okay. Now, let's look at how to do that in RStudio. So again, here is RStudio. I'm going to open up, you would start a new script, right? I'm actually going to open a file so that the code's already done. Okay, so I've opened my script. I have my data set downloaded and in the folder that I want to work in. Then I set my working directory. Right, and I can either do that by session, set working directory, and then choose that directory. Okay, or I can type set wd and then type the path, again using a / on a backslash. Okay. It gets you the same thing. And I should now, in my file window, be able to see all the files I have in that folder. Okay, you notice in this coding here I have 3 hash marks and then some typing. The hash marks or # tells RStudio that that's not code, right? So this is how I annotate the code. So I can put in there what each line is doing without having the code actually get messed up. Okay, so we say that this line of code sets the working directory, tells the program where to find the files and save the files, right? So I've downloaded the file, put it in the folder that I want to work in. Then I've set that folder as my working directory. Now I want to bring in that data file. In this example, we're going to use some data that we collected in class a few years ago. We basically asked students what their home state was and if they called it pop or soda. Okay, the age-old debate. So what I'm going to do is create an object called pop.data, and then less than and dash assign to, and then read.csv. And then I'm going to type in the file name, and that's beverage underscore data.csv. Okay. And then I'm going to run that code, okay, that line of code. Now in my environmental window, you can see that I have a data set, 1,879 observations of 2 variables. Okay. If I want to check that, I can view it. So here's view pop.data, again making sure upper and lower case is appropriate. And here it is. Here I have home state. Notice I have an underscore there instead of a space. Remember, RStudio does not like spaces. And then beverage term, pop or soda. Okay. Now I can summarize my data by typing summary. So if I hit summary pop.data, right, it will say, okay, home state, that's how many observations I have. It's a character class, right? Just another way of sort of viewing it. Now, much like I had with the genetic, sex, and political affiliation, I want to sort of put a table or a tally of my data, because we notice that this is in row form, right? I've got over 1,800 rows. I need to compile that into sort of a summary. How many were from this state that said pop? How many from that state said soda? Okay. I can do that with this table command. Okay. So, table command will summarize data sets in the format in this long version. Okay. So, 1,800 rows. Each row is a sample. Each column is a variable. So I can say table, then I give my data set pop.data, and then $sign. And actually here, let me just redo this so you can see it. So if I go pop.data, $sign, oops. Oh, I was going to screw it up by putting a capital D there, right? Now, RStudio is going to try and finish that for me. I have two variables, home state and beverage term. I want to make so when I make a table, it makes the row is the first variable, the column is the second variable. Okay. So beverage term is going to be the row, home state is the column. Okay. So table pop.data $sign beverage term, comma, pop.data $sign home state. I'm going to run that, and here it spits it out. Okay, so now pop, and each state has its own column. Soda, each state has its own column. Okay, so now I've tabulized my 1,879 observations into this nice little table. I'm going to convert that table into counts. Okay. And this creates a new object in my environmental window. Okay, so it's really just taking this table and turning it into a new object. I'm going to call that object counts. Okay. And now count shows up over here. Okay. It's essentially the same table. I've now just created a new object that is that table. Okay, so I've basically taken my 1,879 observations and I've converted it into an object called counts. And it's this 2x4 table. Okay. Now I want to try to visualize my data, and generally with categorical variables, two categorical variables in a chi-square, we're going to do what's called a contingency table, a mosaic plot, or a stacked bar plot. Okay. And so I'm going to start with the command bar plot. Then open parentheses, then I'm going to tell it what I want it to graph. In this case, I want it to use the summarized data that's in my, what I created as counts. Okay. So I want it to graph this data, this table of data, and I created an object called counts that is this table now. So when I make a table, when I use this line up here, table, it just output it as a table, but it didn't save it as an object. Now when I say counts is assigned to that same string of code, now it creates a new object that is that table. So I have my full data set, but I'm actually going to use the tableized version to make my plots, okay, and to do my chi-square. And that is now called counts because that's what I named it, right? I could name it anything I wanted, and then assign to, is that less than and dash, right? And then table, oops, and then my two variables. So bar plot counts, comma. So this is telling RStudio to make a bar graph from the counts data, or the object that is counts. Main equals, this is going to be my main label. Beverage term versus home state has to be in quotations. Okay. XLab equals This is the label for my x-axes. Remember, we always want to have our axes labeled correctly. Okay, xlab equals home state, again in quotations because it's text. Now I want the color to equal C concatenate, and I've got two terms here, pop or soda. Okay. RStudio by default assumes you're doing it in alphabetical order. So if I wanted pop to be red in pop versus soda, pop would come first, so I'd type red first. In this case, dark blue is going to be pop, red will be soda. So if you had 3 levels, I would do another color, right? If I had pop, soda, and I don't know, Coke, right? Down south they call it just Coke, right? So if I had another category, I would add a third color, and that would actually be listed first because it would Coke, pop, soda, because RStudio does it in alphabetical order. Okay. Again, in parentheses here, if you want to know what options of colors you have, you can use this. It's just colors, open, close parentheses. If I run that, here's all the colors you can use in base R. So what's in quotations is what you would type. Light blue 4, light coral, right? Lots of different options. There's no shortage of color options in RStudio. If you type it out correctly, you should see that the dark blue here comes out in that color. If I misspell it, notice that goes away, right? There, dark blue. But it shows the color if I've typed the color correctly. Okay. And then comma, and then legend. So I need to tell it what to make the legend, and that's the row names from count. So that's pop and soda. Close that parentheses and close the whole parentheses. And now I'm going to run this. Boom. So here's each state. Soda in red, pop in dark blue, and a stacked bar graph or mosaic plot on the proportion of pop versus soda in each of those states. We can see that in Minnesota and North Dakota, most of the people use pop. South Dakota and Wisconsin, it swings towards soda. Okay. That allows me to visualize my data. Now I am actually going to conduct the chi-square test. And that's pretty simple. It's just one command, chi-sq.test. Okay. So chi-sq.test, open parentheses. Then I want to give it my X and Y variables. So pop.data, $, beverage term. So whether they call it. So we want to know if whether you use the term pop or soda is influenced by where you're from, right? So in this case, the beverage term is the dependent variable. The home state is the independent variable. Okay, so we list the dependent variable first, comma, independent variable, comma, and then we type correct equals false. That's just to tell RStudio not to do an automatic correction. In some cases it matters, in some cases it doesn't. If I run that line of code, here it spits out down here. So the chi-square is 761.88, degrees of freedom is 3, the p-value is less than 2.2 times 10 to the negative 16. Okay. Another way to do it is just to do chi-sq.test and counts and forget all this stuff, right? As long as you have counts done the same way or set up correctly, it should do the same thing. This does a correction. Again, in some data sets, a correction changes things and sometimes it doesn't. Generally, if you have a small sample size, the correction makes a difference. We have a large sample size, but it spits it out here. Again, our p-value is far less than 0.05, right? So with a small p-value, we would say, so our p-value is less than 2.2 times 10 to the negative 16th. Therefore, we can reject the null hypothesis and say there's evidence to accept the alternative hypothesis, which states that where you're from or your home state does influence whether you use the term pop or soda. Again, looking back at our visualization here, we would say that if you were from Minnesota or North Dakota, you would traditionally say pop. If you're from South Dakota or Wisconsin, you're more likely to say soda. Okay, that's a chi-square in RStudio.